And besides dietary restriction, are there any other methods to activate the tubular lysosomes and promote lifespan extension, such as medication or some other?
Yeah, so we know we've done some genetic studies, and we know that at least inhibition of a gene called mTOR is sufficient to induce these tubular lysosomes even in well-fed animals. So it sort of mimics the dietary restriction effect. And so mTOR is a major nutrient sensing protein in our cells. And so there are drugs that are targeting mTOR like rapamycin. And so we haven't directly tested that in our system, but we suspect that, things like rapamycin would also induce tubular lysosomes. And we also, found another genetic way to induce tubular lysosomes constitutively. And we can do this by overexpressing another small gene called SFIP. And when we do this, we're able to create these networks, all the time, even if animals are really well fed. And so one of our, long-term goals is to try to target SFIP as a therapy. And one of the kind of cool things about SFIP is that it's a very small protein. It's only 77 amino acids. So it's a very, very short peptide, which is something that could, in theory, be used to synthesize a small peptide that could be delivered sort of like a small molecule drug.
And Dr. Adam, could you please elaborate on the aspect
Yeah.
protein?
Yeah, so we're really excited, I think, about SFIP, and so this was research that Alyssa had started looking at SFIP during her postdoc, and it was studying it because there was evidence from the fly system that this was a key regulator of the tubular lysosome network in this animal. That if you got rid of it, that the tubules didn't form as well, and if you added it back, the tubules just formed very well, very strongly. And this correlated with physiology, that the animals that were lacking SFIP seemed to be a little sicker, showed some degenerative phenotypes, whereas those that had SFIP added back seemed to be very robust. And so one thing that was curious when we started doing this with the worm is we obviously wanted to test in the C. elegans nematode if SFIP could also have a conserved role in regulating these lysosomes. The thing is, though, if you look at the genome of all the genes that are listed for C. elegans, there is not an annotation for an SFIP gene, okay, what we would call a homolog of the fly SFIP. There's not anything that's been identified in the worm to be an SFIP gene. So we could not manipulate that gene in the worm system. Instead, what we did was we added back to the worm system the fly gene. Okay, so what we were doing was we were taking the gene that encodes for SFIP from the flies, and we were having the worms now make this protein. And when it made this fly protein, that's when we saw that these tubular lysosomes were constitutively induced. What we now know is we can take the human version of SFIP and also make the worms express that. And we have some encouraging evidence that I think this is also inducing tubular lysosomes in the animal. And so, yeah, we're really excited to try to see if there are any really key parts of SFIP that are stimulating this, because even though it's a very small protein, if we can get it even smaller, it may be more powerful as a drug or amenable as a drug to help.
It targets it.
Yeah, to do that. So we're trying to understand what parts of SFIP have these capabilities, and if we can, potentially develop it further. If not, there are certainly really interesting aspects of biology that we can learn from it.